Okay, so I'm going to continue this lecture 14, the second part. So we talked about gherkin and torpedo. That's how they are making dorsal polarity of the eggshells. So I'm going to switch to presenter mode. And so we're going to talk about next set of genes. Okay, so those genes I listed in here works for follicle cells, okay? And I think that the next one is this genes that works in the germline, gastrulation defective, snake, and Easter, and Spetzel. There's a second set. And then the first set is probably tall, tube, pale, cactus, and dorsal, how they are working. So we mostly focus on these first, and then I'm gonna move to our next group. Okay, so I, explained to you the dorsal is the single most important gene. They are specialized in ventralizing the embryo, so they are forming localization in the ventral cells. So this is actually a gradient. I mean, I think that here is high, and then as they go to the laterally, then here is the low. So dorsal protein or message is not on the dorsal side of the embryo. It's kind of confusing, right? But what will happen is actually they all have their dorsal protein, but the I think there's cells in the ventral side, the dorsal protein somehow escape the degradation. So that's why they will stay and then form this kind of gradient. On here, they are not dorsal protein on this dorsal side. It's not going to be translocated. I think that's you might want to check with the last slide. So they're all going to be a target of the degradation. So it will be just disappear in the cytoplasm. So when they are dorsalized, means that the dorsal protein is not there. So that's why they are all dorsalized. Okay. And then ventralized means that, somehow overexpression or like a dominant mutation occurs. So let's see here. So that is, nuclear staining of the embryo and then amount of dorsal showing. As you can see, the lighter staining means that less, amount. And so in ventralized embryo, regardless of which axis it is dorsalized everywhere. So it's going to be all ventralized. So dorsalized embryo is the mutant, and then there is the tall. I think that you always have to use as a tall is the capital. I think it was a German word, means cool or something like that. I'm not gonna say too much about German, okay? So when tall is minus, which is recessive, okay? Then they are, dorsalized. And then tall is dominant. So tall is the receptor. Somehow they are hyperactive, or cactus is minus, okay? When cactus is the gone, okay? So I would say this is like a plus, okay? And then they are minus, then they are ventralized. So, tall activate the dorsal nuclear import, and then cactus is blocking it. So cactus is, I would say, negative regulator of the this signaling pathway. So if you remove negative, then it becomes positive. So that's why it's the same as the dominant allele of this. Okay, so tall, dominant tall, or F's, I would say cactus is absent, okay? And then you will see this kind of ventralized embryo that you are seeing. Okay, so toll receptor is not localized, means that it's not active or somehow. Then you take the, so this is, you know, perivitelline space. From that, they took the perivitelline fluid, okay, and they are injecting into here. So injecting into a recipient. And if toll is not there, toll is a receptor, right? So I think that they are trying to get the ligand out of it. So ligand is the, although ligand is here, but toll is not localized or activated or properly distributed. So you don't see any localization. So that's why you will see dosalization occurs, okay? But the ventralization of toll dominant is that regardless of the ligand here, toll is always active. I mean, think about the mutation that somehow switches to the on position all the time, and then you cannot turn it off or something. Then no matter what, they are all active, so they all become ventralized. Okay, okay. And then when toll is properly localized, means that toll is only active, in here, present, and then ligand is somehow only localized to ventral area. Then what will happen is that, this dorsal signals. This is the dorsal, okay? Dorsal distribution is only active in ventral side, so you have a normal embryo. So toll signaling is inactivate cactus. So you will understand that. So there's a ligand here, which we are going to talk about a little bit later, but ligand will bind to toll, and then toll is activated. Then it will activate the, it will go through tube first, and then it will activate the PEL. So PEL will be activated, and then PEL is the fast, kinase. So it will activate dorsal and then cactus first. But when cactus is phosphorylated, and it will be degraded like this. So dorsal will be released, and then they will be jumping into the nucleus. That's what we call translocation. Okay, right. So translocation occurs, and then they will activate downstream molecule. I mean, that detail is not in here, but dorsal, once they go to nucleus, and then I assume that they'll bind to DNA and then activate the transcription. Okay, so that was easy. But the one thing that we need to know is the cactus. Cactus is the off switch. Okay, right. So you need to turn that on. What you need to do is that you need to get rid of the off switch, so that signal can pass through to the downstream. Okay, so next is the gastrulation defective, SNAKE and Easter and Spessel, those genes. Okay, so those genes are mostly about the ligand gene. So toll ligand is in the perivitelline fluid, which is the perivitelline space. So this is the needle. You suck it out, almost some of the fluid, and then this is a PEL, SNAKE, and GD mutant. Okay, but not the toll mutant, right? So embryonic genes are okay, and then you have any mutant embryo here, and then you inject into probably surround here. Then, right, so they are injecting to ectopic place on the dorsal area, and then you are able to turn on dorsal on this dorsal side. Okay, I mean, they try to show this is the ligand, and then the ligand binds to the toll, and then they'll activate the dorsal. So this is only that purpose, right? So you can inject anywhere. So I mean, if you inject into this area, okay, then it's not really surprising, isn't it? So that's why scientists choose a different area and then show to demonstrate the function of this ligand. Okay, so from that way, they find the Spätzle. Spätzle is the German noodle. This is like I took this picture from, I went to Fredericksburg, Texas. And Spätzle is the gene name, and then it's a novel secreted protein. And then Spätzle protein purified from embryo can activate the toll in the injection assay. We have seen it. And Spätzle protein is processed to a small form that is active form. So actually they are bigger form, and then they cut corner and then become active. So because of this fact, okay, next story is that, okay, they wanted to ask, what is processing, processing this Spätzle. So they raised the antibody against the Spätzle, and then they ran the protein extract of each mutant. So this is wild type control: Spätzle, noodle, pipe, GD, snake, Easter, toll, all this mutant, and then they did a protein extract and they ran it. It's kind of easy. You always have to put some embryo into the buffer and then boil them, and then you will get this kind of extract. So you need to have an anti-Spätzle antibody. Okay. So this is the Western blot result. And then they try to detect the Spätzle, where the Spätzle is, okay? And then the Spätzle is, corner is cut and then it will be active. So this is the processed form, okay? So if you see various mutant, and then, but the toll mutant 2 pale dorsal, those downstream gene, even if they are mutant, but they are still able to produce this Spätzle processed form, okay? But here, Spätzle, of course, Spätzle does not produce any Spätzle, right? But noodle, pipe, GD, snake, and Easter, there's 5 genes, okay? When they are missing, Spätzle, precursor Spätzle is produced, isn't it? Right. But the processed form is not there. Okay, right. So from that, they figure this one out. So actually, it's kind of like a protease cascade. So GD is somehow tethered to the ventral eggshells, follicle cell, and then that is activating snake. So that is the protease. So cut some corner of the snake. So snake is active. Then active snake will activate Easter, which is the another protease. And then this Easter, when Easter is activated, and then they'll cut corner of the Spätzle when this is the it's processed, and then they are active form, so that they can bind to this toll receptor on the surface of the oocyte. Okay, so this processed toll, Spätzle really binds to toll. So here, so they're going to be localized to this ventrally, right? So they'll bind to the ventral cells, which has a toll receptor. And then as they go up, probably especially concentration is also pretty low. So that's why they have a less signal. So you have a less amount of dorsal protein going into the nucleus, isn't it? Let me do it again. Especially is on the ventral, and then mostly in the center is the high. As they go through the laterally, it's a lower amount. So Toll receptor is receiving this sparsely, and then amount of signal also form this kind of gradient. So those are protein that go into the nucleus also will have a same kind of gradient, kind of like a property. So lateral cell have less dorsal protein going in, or no dorsal protein going in. So anyway, this is the ventral side. So concentration of dorsal in nucleus is high. And then as they go to, this is a lateral center and very, very low here. Okay, and here's the high. Okay, so I think I have finished with the, chapter, lecture 14. And I'm going to continue on lecture 15 next time. Thank you.